part of the bioinformatics lecture, so we will discuss about biological databases. So, the GenBank, the National Center of Biotechnology and Information, also known as the NCBI, of the NIH in Bethesda, California. So, first is GenBank, and then the next one is DDBJ, which is the DNA database of Japan, at the National Institutes of Genetics in Mishima, Japan, and the European Molecular Biology Laboratory, or EMBOL, nucleotide sequence database at the European Bioinformatics Institute in Kingston, England. So these are the three largest repository of DNA information. So when I say largest, these are not the only DNA repository. There are actually many different databases, but most of those smaller databases usually have specific specialization. For example, the yeast genome database, which is a repository mostly of yeast genomes, yeast genes. But GenBank, DDJB, and EBI, they usually specialize in all other, it's a more of an umbrella for many different organisms. So these are the three largest biological databases, and where those servers are usually located. Now, the GenBank, which is the database of the most known nucleotide and protein sequences. Actually, this is considered the largest. So, it's a database consisting of most known public DNA and protein sequences. So, public, that means it's freely accessible, freely available. It contains bibliographic and biological annotation. So, if you look at the GenBank, you will see there not only the sequence, of course, the details of that sequence, but also links to the studies of the studies where those sequences are first discovered, or where those sequences are originated. So the data from GenBank are available for free of charge from the National Center of Biotechnology Information in the National Library of Medicine at NIH. So you can just actually, I believe you're already familiar with NCBI from your previous courses, so you can actually do that. Okay, so the GenBank, number of organisms in the GenBank, it contains over 260,000 different species are represented in GenBank, with over 1,000 new species added per month. So it's continuously increasing. So this is the data from 2008. So actually more than 10 years ago. So I believe the size of the database have increased significantly. And then, to help organize available information, each sequence name in a GenBank record is followed by its data file division and primary accession number. So, if you look at GenBank data, so this is the basic information for a GenBank entry. So, you have your primary accession number. So, the following codes are used to designate the data file division. So, PRI, primate sequences, ROD, rodents, MAM, other mammalians, VRT, other vertebrates, INV for invertebrates, PLN for plant, fungal, and algal sequences. We have BCT for bacterial, VRL for viral, PHG for bacteriophage sequences, and others. For example, synthetic sequences, unannotated sequences, which is, there's a lot of unannotated sequences actually there. EST, express sequence tags. We have patent sequences. These are, for example, intentionally mutated sequences and patented by industries, usually used for industries. GSS, we have genome survey sequences, sequence tag sites, we have high throughput genomic sequences, etc., etc. So these are usually where those are originated. So types of data in GenBank. So the types of sequence data in GenBank and other database belong to human beta globin. For example, in human beta globin, so when we say globin, it may refer to a gene or any other DNA feature, and an nRNA or an nRNA transcripts, or is complementary, corresponding complementary DNAs or cDNA, or its protein. So we have, for example, a certain entry in NCBI, in GenBank entry, you have either the DNA, the gene sequence with the introns, the cDNA sequence, the mRNA sequence, or the protein sequence. So there are lots of links from that, so you can see actually this. Actually, it's better if you explore it for yourself. So the NCBI creates a public database, conducts research in computational biology, develops software tools for analyzing genomic data, and disseminates the biomedical information. So actually NCBI is a database of databases. So inside NCBI, you have the GenBank, you have the protein database, you have your what other database? For example, OMIM databases, you have your actually there's a lot of different databases in NCBI. So we have Pubmed, actually another database found in NCBI. So it's a search service from the National Library of Medicine that provides access to over 18 million citations in Medline and Medline and other related database. Basically, this is similar to Science Direct. So PubMed is a database of journals, literature involving biotechnology, sequences, bioinformatics. Not only bioinformatics, actually research specializing in medicine. So we have the PubMed, and it's free open access journal site PubMed Central. We also have NTRES. NTRES integrates scientific literature, DNA and protein sequence databases, three-dimensional protein structure data, population study data sets, and assemblies of complete genomes into a tightly coupled system. So PubMed is the literature component of NTRES. NTRES is more of a search engine tool. So that is your NTRES. You have BLAST. BLAST is a tool. BLAST is a basic local alignment search tool. Is NCBI sequence similarity search tool designed to support analysis of nucleotide and protein database. It's not actually a database, but rather a tool for comparing, searching the database for sequence similar for information of a similar sequence. Actually, this is usually used for sequence similarity comparisons. So, set of similarity search programs designed. So, BLAST is actually a set of similarity search programs designed to explore all of the available sequence databases regardless of whether the query is protein or DNA. Actually, we have several types of BLAST tools depending on your query. For example, if you want to look for, we can use nucleotide to search for nucleotides or proteins. You can use proteins to search for proteins, or when I talk about proteins, protein sequences to search for a nucleotide or protein sequence as well. We have the OMIM, the Online Mendelian Inheritance in Man, which is a catalog of human genes and genetic disorders. So, created by Victor McCusick and his colleagues and developed for the World Wide Web by NCBI. It contains links to PubMed articles and sequence information. So, OMIM databases. We have Taxonomy. NCBI taxonomy websites include a taxonomy browser for the major divisions of living organisms. It features taxonomy information such as genetic codes and taxonomy resources and additional information such as molecular data on extinct organisms and recent changes to classification schemes. And then you have the structure. So this one focuses more of molecular structure. So the NCBI structure sites maintains the molecular modeling database, which is a database of macromolecular three-dimensional structures, as well as the tools for their visualization and comparative analysis. So MMDB contains experimentally determined biopolymer structures obtained from the protein data bank. So we have student structure resources at NCBI includes the PDBIST, which is a taxonomy site within MMDB, a CN3D, three-dimensional structure viewer, and a vector alignment search tool, which allows comparison of structure. So a similar database is the RCSB, which usually I use RCSB, by the way, instead of the structured database linked to NCBI. But you can also look for that. Now, if you're looking for a gene or a protein sequence, you usually use NTRES. NTRES is more often used as a search tool. So, NTRES. So, you use NTRES gene, NTRES protein, NTRES nucleotide, or Unigene, Uniprot. So, these are more of the catalogs, proteins, or your gene catalog search tools. So basically, that's it for the introduction for biological databases. Now, for a better hands-on information, please see the laboratory activity I uploaded in the Blackboard. So answer the activity there, and submission is due on next week.